the last type of muscle I'd like to discuss today are the smooth muscles. So smooth muscles don't have sarcomeres, but they do have actin and myosin. And instead of Z discs, these actin and myosin filaments are attached to dense bodies. And instead of this like very structured linear assembly of the sarcomeres in skeletal and cardiac muscles where you have the striations, the smooth muscle has more like a vertical and different orientations between the different cytoskeletal units. And this and unique conformation, which I'll show you in it with a picture, really allows a contraction even when they're super stretched. And this is important, for example, in blood vessels, bronchioles that go into the lungs, digestive organs, and the urinary and reproductive tract, because all these areas of the body need peristaltic ways to propel contents through these organs. So basically in the intestine, we need one part of the GI tract to be contracting to push the food down to the next part of the intestine. And then we need the part below to relax. And then once there's some digestion done there, that segment is going to contract and then push the digested food further through the intestine. So I want to show you about how these actin and myosin cytoskeleton are put in different orientations in this smooth muscle, right? So you can see the dense bodies, right, those little circles. But look, they're in different orientations. They're not all parallel like they are in the heart or skeletal muscle. They're on all these different angles, and this really is important for peristalsis. Again, the contraction of like a tube, which we have these smooth muscles are often in the tube-like tissues in the body. And again, we see that has the actin and the myosin that are there, and they usually completely overlap in the smooth muscle. So to stimulate smooth muscle, neurotransmitter is released by the autonomic nervous system. Again, this is not something we think about. It just happens subconsciously without thinking about it. It's automatic. And the receptor proteins are on the entire surface of the smooth muscle cell. So then all these smooth muscle cells can be stimulated at the same time, much in the way that the heart needs to contract. These smooth muscles need to contract all at once, and they use the gap junctions just like the heart does. Anytime multiple cells are going to be contracting at once, we know that there's going to be a gap junction involved. And just like the atria of the heart need to be contracting all at once, all the muscles surrounding, for example, part of the intestine that are going to contract in that one little segment, they all need to contract at once to be pushing the food down to the next segment. And I should also mention hormones also can play a role in activating smooth muscle contraction. So I want to show you how the contraction of smooth muscles is distinct from skeletal and cardiac muscle. So here we have an action potential that is going to be stimulating depolarization of the smooth muscle. So the depolarization is going to open the voltage-gated calcium channels in the plasma membrane. This is going to release calcium, however, to be inside the cytoplasm. However, what's unusual is there's no troponin. Instead of binding troponin, calcium is going to bind to a chemodulin, and basically there's going to be a calcium chemodulin complex. The calcium chemodulin complex is going to basically go to this myosin light chain kinase that has a phosphate on it, and it is going to dephosphorylate it. So as the myosin light chain kinase is dephosphorylated by the chemodulin calcium complex, it becomes active. So basically, phosphorylation shuts down the myosin light chain kinase. Once it loses the phosphate, it becomes active. What does this kinase do? This kinase has the ability to basically phosphorylate the myosin light chain. So the myosin light chain, which is the myosin cytoskeletal factor, basically can be phosphorylated, and when it's phosphorylated, it'll be activate the cross bridges, which is involving the myosin head, to facilitate the power stroke and contraction. Okay, so the myosin light chain needs to be phosphorylated to facilitate the myosin head cross bridge activation and contraction. So how does this reaction stop, right? Because you see this myosin light chain that's phosphorylated is facilitating this contraction. So it turns out that smooth muscle cells also have another enzyme called the myosin phosphatase. This has the ability to take the phosphate off of myosin. Okay, so the myosin phosphatase will remove the phosphate from myosin and become an inactive myosin phosphatase because it's already taken the phosphate off of the myosin. And then the myosin will be unphosphorylated, and then basically cross bridges will be inactivated, and there's going to be a relaxation of the smooth muscle. So if you just had part of your intestine contract and push the food down, now that segment needs to relax. So now maybe food products coming from a segment of the intestine above can now go to that region. So this last slide of this presentation compares and contrasts the three types of muscles that I've highlighted today: skeletal muscle, cardiac muscle, and smooth muscle. I consider this figure to be, this table to be extremely important, and I urge you to study this and maybe even blank out the different portions of the column so you could write out what the differences are. So, okay, in terms of striation, skeletal muscle and cardiac muscle are both striated. The smooth muscle is not striated. Okay. In terms of the sarcoplasmic reticulum, it's in transverse tubules. We have transverse tubules in both skeletal muscle and in cardiac muscle. There are no transverse tubules in smooth muscle. Okay. And the sarcoplasmic reticulum is really well developed in the skeletal muscle, less well developed in the cardiac and smooth muscle, but it's there. But the really biggie in this row is no transverse tubules in skeletal muscle, but yet it is there. Transverse tubules play an important role in skeletal and cardiac muscles. Another huge difference is troponin. So skeletal and cardiac muscle have troponin, and skeletal muscle and cardiac muscle have troponin. The smooth muscle does not. Instead, it has chymodulin that binds calcium, and it has a unique pathway to be activating smooth muscle contraction that involves the myosin light chain kinase. So let's look at calcium release. So calcium is released in skeletal muscle from the sarcoplasmic reticulum, right? In cardiac muscle, we have calcium entering the cytoplasm from the sarcoplasmic reticulum and extracellular fluid. And in smooth muscle, from the extracellular fluid and sarcoplasmic reticulum. So the skeletal muscle needs direct nerve stimulation from the central nervous system. The heart has its own pacemaker, which we'll be learning about, I think, next week. And basically, it can contract without stimulation from the central nervous system thanks to the pacemaker. And for smooth muscle, in the absence of central nervous system nerve stimulation, it can maintain tone. And there are some visceral smooth muscles that have pacemaker potentials. And critically, I want to point out that the cardiac muscle and the smooth muscle have gap junctions that allow for the muscles of the heart and the smooth muscle to contract in one unit. And skeletal muscle has no gap junctions. So thank you for your attention, and I hope you enjoyed this lecture comparing skeletal, cardiac, and smooth muscle.